Okay, welcome everyone. Thank you for joining the webinar on SwissLipids, a tool that can support the biological and metabolic interpretation of lipidomic data. So this webinar is hosted by the Analytical Division of AOCS, and I'm Francesca Giuffrida, and I'm actually the secretary of the Analytical Division and the head of the lipidomics team at Nestlé Research in Lausanne in Switzerland. So for this webinar today, we are hosting Dr. Lucila Aimo, who is a biocurator at the Swiss Lipid project of the Swiss Institute of Bioinformatics. Dr. Aimo is a biochemist by training, but she became interested in the field of lipids during her postdoc research at the University of California in Davis, and then at the University of Lausanne. For the last 10 years, Dr. Aimo has been a member of the Swiss-Prot Group of the Swiss Institute of Bioinformatics in Geneva, where she joined the Swiss Lipids project. And we met professionally through the International Lipidomics Society. So the presentation will be recorded and made available online by the AOCS. And if you have questions, please add to the chat, and I will read them, and Dr. Aimo can answer them in the order they are received. So I think that's all, and I would like to welcome Dr. Aimo to begin our presentation. Lucila, the floor is yours.
Thank you, Francesca, and thank you, AOCS, for organizing this webinar series. My name is Lucila Aimo, and like Francesca said, I am a member of the Swiss-Prot Group of the SIB. Next. So the Swiss-Prot Group is one of the 67 groups of the Swiss Institute of Bioinformatics. It is based in Geneva, and it is actually the largest group of the SIB. It has been developing knowledge resources for the life sciences since 1986. I joined 10 years ago, and the team is composed of 24 expert biocurators, 10 software developers, plus IT and support staff. Some of the knowledge resources we develop are UniProt, Rhea, and SwissLipids, which I'm gonna talk about today. Next. So why SwissLipids was created? Next. Well, we were contacted by Gisou van der Goot from EPFL and Howard Riesman from the University of Geneva, who were working on lipidomics and needed help integrating their lipidomic data with biological knowledge. This was about a decade ago, and there were no resources back then to link the lipid analyte that they were getting from their mass spec to a lipid structure, and even less to match it in a pathway. Next. So our approach was to turn the problem around and use the knowledge from lipid pathways to infer which were the possible lipid structures and the possible lipid analytes that one could get in their analysis. Next. SwissLipids was created focusing on the creation of lipids, but by using generalist resources that were already existing, like PubMed for all the peer-reviewed articles, because everything is curated from peer-reviewed articles. We don't carry out any experiments. We curate this from the literature. We use KEBI for all the lipid structures. KEBI is a resource for small molecules. We also use UniProt, which is a resource for protein sequences and functional annotation. So we use this one to annotate all interacting proteins and enzymes. And we use Rhea for all the biochemical reactions or transport reactions. To annotate the lipid metabolism. Okay, next. So today I'm gonna introduce you to Rhea, which is the knowledge base for biochemical and transport reactions. Then I'm going to tell you about UniProt knowledge base. And last, I'm going to tell you about SwissLipids, which is a knowledge base for lipids. Next. So we start with Rhea. Next. This is the URL for Rhea, rhea-db.org. You can also scan this QR code in the corner, left corner. This is the homepage. Rhea, as I said, is an expert curated knowledge base of chemical and transport reactions of biological interest. It is the standard for enzyme and transporter annotation in UniProt, and it uses the chemical dictionary KEBI to describe the reaction participants. Rhea is free. It's freely available for anyone to use. You can search, browse, download the data or query. It has a Sparkle endpoint. It has a programmatic access. You can find more information in the latest publication, or if you next scroll down, next, you can find that it offers several tutorials according to how you want to navigate the website. So next. So as I said, all reaction participants in Rhea are described using the KEBI chemical ontology. KEBI is developed at the EBI. This is the URL at the top. You can also scan the QR at the left corner. If you go to the website, this is the homepage where you can also, it's freely available for anyone to use or download. And you can search for all types of small molecules using the names, synonyms, or chemical structure. Next. So, for example, if you search for arachidonate, you will get a unique identifier, which we call KEBI ID. You can see it there below arachidonate. You have the 2D structure, and you also have chemical structure data. Next. You also, it also offers several synonyms. As you know, lipids can have a diverse number of synonyms. So it is important that we include as many as possible synonyms. But what is important about KEBI is that it also offers a chemical ontology that includes structural classification and roles. So, for example, if you see the second line, it says arachidonate has role human metabolite. So you can search in KEBI for human metabolite, and you, of course, you will get arachidonate as well as all those chemicals that have the role human metabolite. Also in the structural classification, you can see that it has several classifications. For example, arachidonic is a long-chain fatty acid anion. So next. So when searching the ontology, you can see this relation. And if you go to next, long-chain fatty acid anion, next, you will see that this one is a fatty acid anion. Next. And this one at the same time has its relationship to a lipid. So there is this ontology that because Rhea uses this ontology, you can search in Rhea using the ontology. Next. So for example, if you search in Rhea for arachidonate using the KB ID for arachidonate, you will see that you get about 82 reactions. Next. But you can also search in KB using that in Rhea using the KB ID for long chain fatty acid anion, and then you will get 10 times more the number of reactions because you have the 82 from arachidonate plus all other long chain fatty acid anions that are participants. And next, and if you go higher in the ontology and look for the KB ID corresponding to fatty acid anion, you will get even more reactions, 1500. Next. And now if you search using the KB ID corresponding to a lipid, you will get over 6,000 reactions. All these reactions you can download and use in your software. Next. So say, for example, we want to see a reaction that involves arachidonate. We use that KB ID. We get the 82 reactions, but you can also filter those results using the menu on the left, like saying, okay, I only want to see the ones involving proteins, or only the ones that are transport reactions, or those that have been classified using easy numbers. So you can go filtering the results. Let's say, for example, we want to see this Rhea ID, this reaction. We click on it. Next. And each Rhea record has 5 sections. First, you see the 2D structures of the equation, where you can see what are the groups that are changed. All reactions are balanced for mass and charge, so you will see the co-participant or the co-product. Then you have a section for the enzymes. If there are enzymes annotated for that entry, then you will see them in this section. You can also see the description of the reaction participants, which are from KB. So if you scroll down, you can see these participants with all the chemical structure data. This also, you can hide it or you can display it, but also if you want to search through the participants, you have a menu that helps you navigate further, like this one in the bottom. Next. Then the third section involves the cross-references. You have cross-references to different resources like UniProt, easy numbers, gene ontology, KED, MetaPsych, Reactome. And then the last section is the publication, the list of publications where the evidence was taken from. So yes, all the reactions are curated from peer-reviewed publications. Next. So maybe if you are already using other resources like say KEG for the reactions, you can still use Rhea by using the ID mapping tool, which is up here, and you just select you enter the ID of your reaction, and it will map it to Rhea. In this case, for example, for this example, we have one matching reaction in Rhea, and then you can get the information about the enzyme class and the enzymes that have been annotated for that reaction in UniProt. Which brings me to the next, point, that is introducing you to the UniProtKB, that is for protein sequences and functional annotation. Next. So this is the URL for UniProt Knowledgebase, which we abbreviate UniProtKB. It's uniprot.org. This is the homepage where you land. It is freely available for anyone to use, browse, download. It also has programmatic access. As you can see, it has several sections, but today we are only going to focus on the UniProt Knowledgebase, which is the blue square on the left. Next. So UniProtKB entry will have also several sections. First, the biological and functional data, then the sequence and structure data, and then publications and cross-references. Next. So if we look at a UniProtKB entry, you will see, in this case, acid ceramidase, it has a unique identifier. It has the names, the organism, and the function is described with a text. But all this text, we provide the evidence, the source for the supporting evidence. So everything has an evidence tag with a PubMed ID from where it was found. Next. Approximately half of all protein sequences entries in UniProtKB describe enzymes. And before 2019, this enzyme activity was annotated using the IUBMB enzyme classification. So this is the easy numbers and a text line. At the end of 2019, we incorporated Rhea as the standard vocabulary to describe enzymatic activity. And since then, we have been annotating all the enzymatic activity using Rhea. We do still add easy numbers, but Rhea is the primary vocabulary for enzymes. So this way we have both human readable and machine readable forms. Next. If you want to read more about how Rhea is used to describe enzyme activity, you can check out this publication. Next. And now, if you look at UniProtKB entry, you will see this kind of annotation where the catalytic activity is in a text form, but with an evidence tag and PubMed supporting evidence. And it has also next link to, yes. It links back to Rhea. You have the Rhea ID and it links back to Rhea. You have the 2D structures that you can look at in this same entry page, but you can also hide it if there are many reactions. Maybe you only want to see the text. Next. But if you look at the 2D structures, you will see the KEBI participants, and also you can link back to KEBI to search for each one of these. Next. So since now UniProtKB has incorporated Rhea and KEBI, you can now, this means that you can now search UniProtKB for example, lipids, small molecules like lipids. And to do this, you can use several approaches. One is using the UniProt website. Another one is using the UniProt website REST API. And lastly, using the UniProt Sparkle endpoint. Next. So to query lipids using the UniProtKB website, you can just type in the search box the name, the chemical name, or you can use the KB identifiers if you have them, or use the KB ontology, or also you can use structure descriptors such as INCHI keys. Next. You can use the advanced search. For example, in this case, I use the advanced search and chose small molecule and entered next the INCHI key that corresponds to arachidonate, and next, and then search which are the proteins associated to this INCHI key, next. And you can see you get over 12,000 results. Next. So you can also add more fields to your search and say, for example, use the taxonomy and choose next to add the organism human. So this is the ID for human, next. And then you have the INCHI key and the taxonomy, next. And then you reduce the number of results to only human proteins annotated for arachidonate, and this brings you down to 124 results. Yeah, next. Well, another way is to use the website REST API. This allows to access data and tools from the UniProt website with any programming language. And you can retrieve entry result sets using URLs. These URLs can be bookmarked, linked, and used in programs. And let's look at some examples. Next. For example, if you want to look for proteins linked to arachidonate, then this would be the URL where you specify the KB corresponding to arachidonate. Next. But if you only want human proteins linked to arachidonate, then in the same URL, you add the ID for the organism, like in this case. Next. And if you want human proteins linked to any lipid, then instead of the KB ID for arachidonate, you use the KB ID for a lipid, like in this case. Next. Okay, so the third mode of querying UniProtKB would be using the Sparkle endpoint. You can find it at this URL, but you also have in the top menu of the main website the link to Sparkle that brings you to this page where you have the query box. I am not an expert in Sparkle, so I invite you to learn more about Sparkle, checking out the documentation down here, or using the examples that are offered on the right menu. There are over 50 examples. Next. So for example, if you want to retrieve human enzymes that metabolize sphingolipids and are annotated in Campbell. Campbell is a database for chemicals with drug properties that is developed at EBI. Then you use this query and submit it, and you get the result with all the UniProt accessions and Campbell entries linked to it. So the advantage of Sparkle is that it allows to perform complex queries, and not only in the UniProtKB resource, but also across other resources that provide Sparkle endpoints, such as Ensemble, Rhea, or SwissLipids. So next. So now I'm going to tell you about SwissLipids. Next. SwissLipids was designed to facilitate the integration of lipidomic data and lipid biology. Next. So this is our workflow. We curate lipid biology from peer-reviewed literature. First, we curate the lipid structures in KEBI, using KEBI. And then we create the lipid metabolism by using UniProtKB for the enzymes and Rhea for the lipid reactions. And we annotate all these in SwissLipids. Now, next. As you know, many of the membrane lipids are modular structures that have like hydrophilic head and fatty tails. And through the curation process, we can know that there are different fatty tails that can be exchanged for a certain head group. So next, what we do is consider these building blocks and use the different lipid heads. Like in this case, we have an example of glycerol and lipids and glycerophospholipids, but we also have, you can also have sphingolipids that the fatty acid chain can change, and other compounds. Next. And from our curation of the literature, we know many types of fatty acid tails that can exist and be found. And this is just an example, but there are over 100 fatty tails, not only fatty acids, but in some cases it can be fatty alcohols. Next. So what we do is we combine the different possibilities of fatty tails with the different possibilities of lipid heads to generate lipid structures. And this we do by following a combination of rules, because it's not that we can just match any. Okay, so next. And by doing this work, this enumeration, we get over 500,000 lipid structures that are part of our in silico lipidome in SwissLipids. They are linked to KEBI, Rhea, and UniProt. And this is only an example of some of those structures. Next. So after enumerating all these lipid structures, what we do is we organize them in a hierarchical classification in order to provide the possibility of linking to a lipid analyte. Next. So what is a lipid hierarchy? In this example, I'm using phosphatidylcholine, a little bit higher. Can you scroll? Yes, yes. So when we annotate lipids from the literature, we normally know exactly the composition of the structure, like the lipid class, each fatty acid, how it is composed, where are the double bonds, where the fatty acids are within the glycerol molecule, SN1, SN2. Okay, but this is very rare to find in lipidomic studies, where the methods don't allow to know such high resolution of the lipid structure. So maybe, so we have a hierarchy where maybe the method tells you the lipid class and the composition of the fatty acids, but not where the double bonds are. So that would be the first parent here, or you know only the fatty acids, the number of double bonds, not where they are, but you also don't. The method doesn't tell you within the phosphatidylcholine if it's at SN1 or SN2. Or maybe the method only tells you, okay, it's this lipid class and the sum composition. So the sum of the two fatty acids, the total number of carbons, and the total number of double bonds. So we offer this hierarchy so that when, depending on the type of analyte that you have, you can search in the hierarchy. Next. So this is the SwissLipids website. The URL is swisslipids.org. If you go to that website, you've reached this homepage that you have a search box where you can type the name of the lipid or the KB ID and search. But again, like the other resources, SwissLipids is freely available for anyone to use, query, browse, download, and it also offers a Sparkle endpoint. Or you can use other tools like the browsing the classifications. Next. So say, for example, you want to browse by lipid analyte because you have a sample that you run in a mass spec, and the mass chart says indicates it's a PC with an ether bond, 36 carbons, and 4 double bonds. Then you go to SwissLipids and choose to browse by lipid analytes. Next. And this will bring out a menu where you can select the lipid class. So in this case, I use a PC with an ether bond, and then choose the number of carbons and the number of double bonds. Next. And it will browse through the hierarchy, offering the different matching structures. So for example, you have the species would be the PC036-4, and then this one has 9 molecular subspecies, 11 structural subspecies, and 28 isomeric subspecies. But if you already know maybe the different fatty acids, then you can filter down and you will get less number of isomeric subspecies. Okay, so you can see next to each structure, there's a set of colored icons. Next. So this set here you can see more clearly. This set of colored icons indicate the type of annotations that we have in SwissLipids. If they are highlighted, it means there is that type of annotation. So in this example, you can see that specific lipid structure has cheminfo, has classification, and has metabolism annotated. Next. So if we click on that lipid structure, you will see the lipid record where you have the name and a unique identifier. The 2D structure, synonyms, and below you can see a menu. Within blue, you have the different options. Metabolism, for example, we can choose to see the lipid metabolism by clicking on metabolism. Next. It will show you the reaction where that where that lipid participates with the Rhea ID, so it can link back to Rhea. And it also has on the left the enzyme that was seen to metabolize that specific reaction with the UniProt accession. Also, if you click on that enzyme, because you can see which other reactions have been annotated in SwissLipids for that enzyme. And you can also check out the piece of evidence that is supporting this annotation. And the link, it also links to the PubMed ID from where it was sourced. Next. Okay, the other browse option is by lipid class. You can see here in the left the lipid categories that we have in SwissLipids. As you go clicking on each, you can reveal more class members. So if we click on glycerolipids and then in monoradial glycerols, we can see the options. And if we choose one acyl SN glycerol, then you see all the members of that class. And again, you see the set of colored icons indicating the type of annotation that we have in SwissLipids for that lipid structure. Next. Okay, the last tool that I want to show you that you can find in SwissLipids is the identifier mapper. We have cross-references to lipid maps, HMDB, KB. So next, say, for example, you are already using a lipid maps IDs. You can enter these IDs in the mapping box and choose what you want to map it to. For example, if you want to see the proteins, you choose UniProtKB, and the tool next will search in SwissLipids for those lipids that are annotated for the UniProt proteins that are annotated for those lipids. So in this example, these two lipid maps IDs are mapped to 45 UniProtKB identifiers. There's the list, and you can download this list for your use. Next. Okay, so that is mostly what I wanted to show you of the SwissLipids website. We are currently working on boosting the coverage of enzymes and transporters in UniProtKB and Rhea. As you might imagine, the curation work is a very slow process. So we are now participating in machine learning approaches to try and accelerate the curation process. There's a lot of data being published every day. This year we started collaborating with the Recon4IMD consortium in the reconciliation of human lipid metabolic networks. Next. This is just an example of how knowledge bases are used, in this case, to build disease models. Recon4IMD, you can search it up. This is the URL. But basically, Recon4 is a next generation genome scale model of human metabolism dedicated for the inherited metabolic diseases. These affect 1 in 800 newborns. Normally, these diseases are very hard to diagnose. It takes a long time. So the idea is to build and leverage these models to accelerate patient diagnosis and personalize the management of these diseases. The coordinator of this work is Ronan Fleming at the University of Galway, but there are scientists and clinicians from diverse disciplines. We are together with Rhea, UniProt, and SwissLipids trying to cover all the lipid metabolism and the proteins that are affected in inherited metabolic diseases to try and cover all those annotations. Next. And well, of course, the future work we plan to build the next iteration of SwissLipids using UniProtKB as our annotation source. Now that UniProt has adopted Rhea as its standard for enzyme annotation and KB for all the small molecules, we can use UniProt to annotate SwissLipids and design more lipid structures to continue and grow the in silico lipidome in SwissLipids. So far, we had focused mainly on human and mammalian lipids, but since in UniProt we have curators for bacteria, plants, fungi, then we can try and cover these taxa as well. Next. Okay, so in summary, just to summarize, Rhea, UniProt, and SwissLipids knowledge bases are freely accessible and available for anyone to use. They can all be queried for lipids using names or small molecule data, including chemical structures as well as KB identifiers. They all provide a range of search and browse options. They can be downloaded, and they have programmatic access and Sparkle endpoints. And they offer improved support for computational studies of metabolic systems, enzyme function and evolution, omics data, and resource integration. Next. So yes, just to show you the UniProt team is a great part of this work. It not only includes the SIB in Switzerland, but also the peer in the US and the NBL EBI at UK. Next, the Rhea team is much smaller. It is developed at the SIB, managed by Alan Bridge, and with Anne Morgan as the lead curator and developer. Next. And SwissLipids, which is also developed at the SIB, curated mainly by the Swiss-Prot Group, but developed by the Vital IT Group. Next. And if you have searched for your lipids, reactions or proteins, and you couldn't find them in our resources, please don't hesitate to reach out. These are the emails where you can contact us, and you can follow us also in Twitter. Next. And I want to thank you for your attention, and I am open to questions that you may have.
Thank you very much, Dr. Aimo, for the presentation. It was really interesting, and actually I see that from the last presentation you did update a lot and us introduced many more tools in your database. So we have a couple of questions in the chat. I can start reading the first one, asking if it is possible to download selected characteristic of several lipids in order to use the data for other type of studies. For example, for instance, developing new ads and machine learning studies.
You can download the different characteristics, yes, of lipids. If you look into the download section, you will see that you have a file for the proteins, the interacting proteins, another file for the lipid chemical data. So yes, you can download the different types. As for developing new ads, I'm not sure what that means.
New apps. No,
yeah,
the person was referring to new apps.
Okay. Yeah, I guess. Yeah, you can use it's freely available. Yeah.
And then we have the person, thank you for the answer, Lucila. And then we have another, I don't know if it's a question or more a statement, saying that the use of artificial intelligence for structural identification of macromolecule or their activities can provide more precision on all the molecular interaction in collect of bank information. So I'm suppose that this person is proposing the use of artificial intelligence rather than a database, knowledge database, if I understand well the statement. So how do you see the artificial intelligence? Do you think that yeah, it's something that I was wondering as well. If I take arachidonic acid, right, I ask ChatGPT about arachidonic acid and metabolic pathway. How do you think he's going to answer? Would be more I mean, the type of information will be comparable to the Swiss lipid database. How do you see it?
I think one problem with ChatGPT would be what is the source, right? So where it's taking the information from. In the SwissLipids database, you can know exactly where that information is coming from, and you have the links to the publication, and all the publications are peer-reviewed articles. So I think I would be more confident to trust Knowledgebase than ChatGPT, to be honest.
Yeah, I think, I mean, you can always ask ChatGPT, but then please double check with the Swiss lipid database, because as Dr. Aimo was saying, yeah, the information is, yeah, it's checked, evaluated. And I have a question about it. I mean, I see that you're a team of creator, but how does it work? I mean, I suppose you have a system that tell you when a new article, new paper is out, and then how do you proceed? Do you read it in, let's say, a couple of people, in more people, to be sure that the science behind is strong enough for being updating your database? How do you do?
Yeah, that's a good question. Yes, normally at least 2 curators read the paper. One makes the annotations, but the annotation needs to be approved, and this needs to be done by a second curator. So yes, the annotations are doubly checked, and if there is any discussion, maybe a third curator is invited to discuss more, or the whole group is invited to the discussion. So yes, at least 2 curators for the annotations.
Okay. And I was also thinking about, I mean, there are many databases, right? I'm an analytical chemist, so I have the tendency to use a lot of lipid maps. How do you see the competition between your Swiss lipid database and other databases like lipid maps?
Yes, well, yes, lipid maps is a really good database for lipids. I think it started more. I think it's very useful for the analytical side because it offers many tools like MS tools and lipidomics tools, but it was actually lacking the link to the lipid biology, to the lipid metabolism, to the enzymes. And that's why these researchers in Switzerland contacted us because that was missing. That's why we started SwissLipids. Yes, I know now, like a year ago or so, Lipid Map started adding information about lipid metabolism, and it actually links back to SwissLipids and uses Rhea as well. Yes, but I guess now they have only one lipid curator, and UniProt having 24. I think we can provide much more information on lipid metabolism in our resource. So yeah, it depends on the aim. Also, I think we have this lipid hierarchy. So depending on the lipid analyte that you have, you can browse our lipid resource using the hierarchy, which I don't think Lipid Maps has.
Thank you, Dr. Aimo. I don't know if there are any additional questions. I don't see any more in the chat, but please feel free to write down any comment, any question. We still have 5 minutes time for a last burning question. So, Dr. Aimo, you were saying that as next step, you will also focus on disease, because if I understood well, at the moment you have, I mean, let's say standard metabolism pathway, right, in the Swiss lipid database. So that will be another chapter, like another chapter of the database. How are you going to?
Yeah, well, the disease will be mostly on lipid metabolism, so we will annotate the reactions involved in Rhea and see if the enzyme is catalyzing that we annotated in UniProtKB. So they will all be linked. So it's not, so yeah, you will find in SwissLipids later on. But yeah, we will focus on annotating the enzymes in UniProtKB using the reactions from Rhea, and SwissLipids will mostly link back to them.
Okay. Okay. Thank you. Really clear. So if there are not other questions, I thank you really much for the presentation. It was really interesting from my point of view. I hope that the audience as well found it of interest. And thank you everyone for joining us. And I wish you a nice rest of the day if you are in US, or the evening if you are in Europe. And yeah, stay tuned on next webinars in AOCS. Thank you very much.
Thank you.
Bye-bye.
Bye.